welcome to the deep dive. Today we're looking at a truly groundbreaking idea in cellular health, aging, maybe even how we tackle genetic disorders.
Yeah, it's this idea of sidestepping some, fundamental biological limits.
Exactly, like a kind of cellular re-engineering. That's what we're hoping to unpack in this deep dive.
Right. We're focusing on a new way to handle issues with mitochondria, like the powerhouses in our cells, instead of trying to fix their DNA directly, which is, well, tricky.
Very tricky.
The idea is moving the genetic instructions somewhere else, somewhere safer in the cell.
Right. And you, our listener, you're looking for a solid grasp of this stuff quickly. And that's what we aim for. We're digging into work from the MitoSense Project, the Longevity Research Institute, Lifespan.io reported on it.
Yeah, good sources.
So the mission today, understand this new frontier, expressing mitochondrial genes from the nucleus and what that could actually mean for health, maybe even longevity. So let's get into it.
Okay, so first, to really get why this is a big deal, we need a quick refresher on mitochondria.
The powerhouses.
Exactly, the powerhouses. It's a good analogy. They generate almost all the energy, ATP. Our cells need to, do anything.
Through that electron transport chain thing, right? Like a molecular bucket brigade.
Kind of, yeah. A series of handoffs generating ATP. And almost every single cell you have, except red blood cells, relies on these little factories.
And they're unique, aren't they? There's something special about their setup inside the cell.
Yes, exactly. Mitochondria have their own DNA, a separate small set of genes called mitochondrial DNA, or mtDNA.
Right, left over from their bacterial ancestors, I remember that.
Yeah, it's a relic of their ancient bacterial origins. Now, evolutionarily, that independence was useful, but mtDNA has a big weakness. It's really quite vulnerable to damage.
Vulnerable how? Like more than our main DNA in the nucleus.
Oh, much more. See, the DNA in our nucleus has all these sophisticated repair systems. mtDNA, not so much. It doesn't have that level of backup. So over time, it just tends to collect mutations, errors, glitches.
And that's where the problems start.
That's where it gets really relevant to aging and disease. These accumulated mutations mean mitochondria don't work as well, energy production dips, and this decline, it's being linked more and more to aging itself and, degenerative diseases too.
Like Alzheimer's, Parkinson's.
Exactly, those, and some muscle disorders too. Think of it like your cellular power grid running on software that gets buggier over time and is hard to patch.
Okay, that makes sense, like a machine wearing out. But the repair crew isn't well equipped. And this isn't just an aging thing, right? People can be born with mitochondrial issues.
Precisely. There are inherited mitochondrial diseases. These are caused by faulty mtDNA passed down, and they can be devastating right from birth sometimes.
Wow.
And what's really tough is that our current gene therapy tools, the ones we use for fixing genes in the nucleus, they haven't really worked for fixing mtDNA directly inside the mitochondria. It's a different challenge.
Okay, so that really sets the stage. We've got these vital powerhouses with vulnerable DNA that's hard to fix directly. So tell us about this clever workaround you mentioned, moving the expression.
Right. This is where allotopic expression comes in. It's basically a strategic relocation.
Relocation.
Yeah. Instead of battling to fix the fragile mtDNA inside the mitochondria, researchers are trying to take the instructions, the genes themselves, out of the mitochondria and move their expression to the nucleus, the cell's main control center, which has much better repair systems, better regulation.
So you're not fixing the original faulty blueprint in the mitochondria.
Sure.
You're making a new, clean copy and running it from the main, safer factory.
Exactly. It's like moving critical manufacturing to a more secure facility. And this is what the MitoSense and Longevity Research Institute teams did, the work Lifespan.io covered. They successfully recoded a mitochondrial gene and got it working from the nucleus in mice.
In living mice. Wow.
Yeah, in live animals. It's a big step.
That sounds huge, but I imagine it's not just drag and drop, right?
Yeah.
It can't be that simple.
Oh, definitely not. Huge technical hurdles. For starters, the genetic code isn't identical. Mitochondria use a slightly different dialect, you could say, than the nucleus. So the gene has to be carefully rewritten, recoded, so the nucleus machinery can read it properly.
Okay, like translating it.
Exactly.
Yeah.
And then there's the protein itself. Many mitochondrial proteins are hydrophobic. They don't like water.
Right.
Which makes moving them through the watery cell and getting them back into the mitochondria pretty tricky.
Okay, so it's like making a special part in one factory, but then you have to ship it carefully through a difficult environment to where it's actually needed in another factory. How did they solve that delivery problem?
The key is something called the mitochondrial targeting sequence, or MTS.
MTS.
Think of it like a zip code, or maybe a specific shipping label, a molecular postcode.
Huh, I like that.
When the protein is made in the main cell body, based on the instructions now in the nucleus, this MTS tag is added. It basically tells the cell's transport systems, hey, this protein belongs in the mitochondria.
So it guides it back home.
Precisely. Ensures it gets imported back into the mitochondria where it needs to do its job.
That is clever, like a built-in GPS. Okay, so this specific research focused on one gene, ATP8. Why that one? What's special about ATP8?
Well, ATP8 is crucial. It holds the instructions for one part, one subunit of ATP synthase.
And ATP synthase is the...
Yeah, the final machine. The amazing molecular motor literally spins at the end of the energy production line that actually makes the ATP. The energy currency.
Got it. The thing that actually cranks out the power.
Exactly. So if ATP8 is faulty or missing, that whole ATP synthase complex falters, energy production tanks. It causes a real cellular energy crisis.
Okay, so messing with ATP8 really hits the cell where it hurts, which makes the success here even more significant, right? The protein made from the nuclear gene actually worked. It fixed the problem in the mice.
That's the really exciting part, yes. The protein made from the nuclear ATP8 gene got shipped back to the mitochondria, it slotted into the ATP synthase complex correctly, and it restored the complex's function. It started making ATP properly again.
Okay.
It's a powerful proof of concept that, yes, this strategy can actually work in a living animal.
Okay, so proof of concept in mice. Thinking bigger picture. What are the implications if this could work for other mitochondrial genes and maybe down the line in people?
Oh, the potential is really vast. I mean, first, think about those inherited mitochondrial diseases we mentioned.
Yeah.
Instead of the near impossible task of fixing the bad mtDNA, maybe you could use gene therapy to deliver a working copy of the gene to the nucleus. Just bypass the faulty mitochondrial gene altogether.
That would be life-changing for those families. Absolutely huge.
Totally revolutionary for them. And then there's the aging angle.
Right, the general decline in energy.
Yeah. We know mitochondrial function tends to decline as we age, partly because of that accumulating mtDNA damage. So maybe allotopic expression could, revitalize aging tissues, keep that energy supply robust by providing functional proteins from the nucleus.
So potentially counteracting some of that age-related slump we feel.
That's the hope. It might not just be about treating specific diseases, but maybe boosting our resilience to aging itself.
Making our cells fundamentally stronger.
Sort of, yeah. By moving these vital instructions to the safer nucleus, maybe cells become more resistant to mitochondrial problems caused by stress or toxins or just time. It's about potentially bolstering the whole system.
This sounds incredibly promising, almost too good to be true. What are the catches, the limitations? Where does the research need to go next?
Well, it's still early days, relatively speaking. This success with ATP8 is fantastic, but we don't know if all mitochondrial proteins can be easily moved like this. Some might just be too difficult to express properly in the nucleus or too hard to import back into the mitochondria, their structure maybe, or other properties.
Makes sense.
And crucially, we need a lot more research on long-term safety and efficacy, especially before even thinking about humans. Are there any, unintended side effects of having these proteins made in a different place over years or decades? We just don't know yet.
Right. Safety first. Long-term studies are key. So a major breakthrough, definitely, but the path to any kind of therapy still has a way to go.
Absolutely. But getting it to work in live mice, showing that restored function, that's a massive hurdle cleared. It suggests this very bold idea is actually feasible.
Yeah, it moves it from science fiction towards potential reality.
Exactly. It makes you wonder, what other fundamental biological limitations could we maybe engineer our way around with clever thinking like this?
That's a really powerful question. Okay, so let's just quickly recap this deep dive. We've looked at research from Mitosense and the Longevity Research Institute, reported by Lifespan.io, showing they successfully expressed a key mitochondrial gene, ATP8, from the nucleus in live mice. This whole strategy, allotopic expression, gets around the problem of fragile mitochondrial DNA.
Right.
And it opens up really exciting possibilities for treating mitochondrial diseases, maybe even fighting age-related energy decline.
Yeah, it's a big step forward in engineering cellular resilience, you could say.
Cellular resilience, I like that.
Giving mitochondria a better blueprint, basically, from a safer place, it tackles a core vulnerability in our cells. It's a bold idea, kind of an evolutionary hack, really,
Yeah.
with potentially huge implications for medicine and, well, longevity science down the road.
Truly fascinating stuff. This definitely gives you, our listener, a new way to think about cell health and maybe the future of medicine. And it leaves us with that final thought to chew on. If we can effectively relocate and run these essential genetic programs from a different place, what other biological limitations might eventually be open to a bit of cellular re-architecture? Makes you think.
It certainly does.
Thanks for joining us for this deep dive.